All right. Well, I think we have a really nice group started. So, without further ado, you're certainly here to not listen to me, but definitely hear from Emily Grave, our featured speaker for today. So, for those of you who I haven't met yet, my name is Christina Graham, the superintendent of Honouliuli National Historic Site. And on the call with me in the background is Wes, who is with the Pacific Historic Parks, which is the park's great nonprofit organization and partner, which is hosting this wonderful virtual speaker series. And Wes is behind the scenes making sure that we're all enjoying Emily's talk together. So thank you very much for Pacific Historic Parks. So some of you may wonder if you have familiarity with the park and its focus. It is the largest and longest used Japanese incarceration site in Hawaii during World War II. So you might be wondering, why are we talking about a native species, the Wiliwili? Well, part of the park's responsibility is also to manage not just for the historic and cultural resources, but also for the natural resources. And when I first started in this position, I was really excited to hear that we actually have Wiliwili in the park, which is a very rare dry lowland forest species. And so my background actually is a lot in native habitat and conservation work, especially here locally. And so I was really excited to learn that we have this really rare species here. And so this month we are dedicating to the native species at the park. And I'm really excited to be able to introduce Emily to you. She is the senior manager of forestry partnerships and sourcing at Terraform Nation, where she leads efforts to promote sustainable forestry practices that help combat climate change. With a strong passion for conservation, Emily has focused her career on ecosystem restoration and building environmental partnerships worldwide. She earned a Master's of Science in Botany and a Bachelor of Science in Ethnobotany from UH Manoa, where she gained extensive knowledge in plant sciences and the ways in which people interact with plants. So, without further ado, I will hand it over to Emily to share more about her work on Wiliwili. Thank you so much.
Thanks, Christine. That's a lovely introduction. And hi, everybody. It's nice to see some names that are familiar. Hi, Dad. And all these Willy Willy enthusiasts. My name is Emily Grave, as Christine mentioned. I did my master's thesis on the endemic Hawaiian coral tree. That's Willy Willy. You may see me looking that direction, and that's because the slides are on that screen. So just a little housekeeping there. And some of these slides are directly from my defense presentation. Others are brand new. This tree has absolutely captivated me for years. I always wondered, why can I only see them in certain places? Why do you have to decide to go and see the Willy Willy? Why can't you see them everywhere? Because the habitat is so extensive. It seems so, right? So I started looking for the answer to that question during my first year of grad school at UH Manoa. And here's a little bit of what I found that first year. So dryland forests globally are the most imperiled of all, and the trend is, of course, consistent in Hawaii. Wili Wili is a dry forest species, and that's actually where 25% of officially listed endangered Hawaiian plants are found. So we've lost roughly 90% of dry forests over the last 2 centuries, but why? Why is this happening? In dry forests, you can see in this picture here, destructive invasive grasses like Panica maximum that make excellent fuel for fires and literally cover the landscape, shading out any kind of potential for new recruits. There's also land development that's not very conducive to preserving habitat. Introduced animals, right? We've got pigs, sheep, goat, deer, and then we also have two invasive arthropods that have had a catastrophic effect on one species in particular. That's Erythrina sandwichensis. And those are the Erythrina gall wasp and the bruchid beetle, as you might have guessed. So Willy Willy is just one of those iconic species, stands out in the middle of the forest. Um, and it was a dominant dry forest tree found on all 8 of the main Hawaiian Islands. So Willy Willy is a summer deciduous tree. It's one of our only deciduous trees, actually. It's an early adaptation to these dry systems in order to conserve the water and resources, right? They gain their leaves in the wet season and they shed them right before flowering in the late summer to early fall. Those swollen trunks are conspicuously colored orange and yellow, right? Almost just like the flowers. And of course, the infamous prickles. They are not thorns, they are not spines, they are prickles. And if you want to wax intellectual on that subject, please call Dr. Cliff Morden at, no, I'm just kidding. But Wiliwili demonstrates this phenotypic plasticity all across the main Hawaiian island chain, where trunks on Kauai are more smooth. And then as you move south down toward Big Island, the trunks are completely covered in prickles. So that's actually what piqued my curiosity around the genetics. But the tree is it's culturally important due to its light wood, first of all, right? It was very buoyant, making it excellent for use in traditional items like surfboards and canoes, fishing containers. Temporary lei was made from the flowers, but permanent lei are created from the vibrant seeds. They still are today, right? This was found on Pinterest. And then there's also medicine and charcoal dye they're known for. You know, as an ethnobotany graduate, there's just a ton of history associated with this species. And it seems like so long ago, but starting my love for the tree, it's the ethnobotany that got me, that I chose just to work with this species. So we're going to take a look at some of the lesser known aspects of the ethnobotany today. And some of that is what I was just speaking about, the medicine and the dye. So it was actually the flowers of Wiliwili was one remedy to treat venereal disease. And also another remedy was made from, it was a tea, pounded bark from the Wiliwili that was combined with chili pepper and ava, Piper methysticum, that one. And then in the morning, you follow that with koali. That's a morning glory species. And then for the dye, so the bark is super rich with tannins. And the wood, you can burn it to then make charcoal. It can be used with other color pigment ingredients as well in making dyes for like kapa cloth and so much more. And then with respect to agroforestry, the Wiliwili has an ability to fix atmospheric nitrogen. And it also tolerates strong winds and droughts. So it's used quite often in revegetation programs in highly eroded areas all over Hawaii. So it's frequently planted as a live fence. Also, on account of its readiness, it takes root as soon as it's planted. And again, because it's tolerant of these degraded conditions. And you can use them for windbreaks for the same reason. And then the nutrient-rich leaves, also excellent for soil mulching. But notably, Wiliwili has open crowns like half the year, right? So you can plant other crops underneath it, doesn't overly restrict the light. And also the trees obviously are quite prickly, so they can provide some, impenetrable barriers to protect from unwanted intrusions. So beyond the many traditional uses for Wiliwili, early Hawaiians had a really close relationship with this tree. There's an olelo no eau that links the Wiliwili tree to sharks because Wiliwili's flowering season, it typically occurs in late summer to early fall. And this period coincides with increased shark activity, especially near shore, right? During the breeding season for some species. So Native Hawaiians used these observations to time fishing and swimming and other voyaging activities. It's an example of that phenology-based wisdom where plant cues kind of indicate changes in animal behavior or environmental cycles as well. And that just goes to show the deep connection between the Hawaiian people and their natural environment. Plus, there are multiple species relationships encoded in language and in practice. Like this. So the name Wiliwili, it literally means twisting. And this refers to the twisted seed pods that do this twist as they dry and dehiss. You can see in this picture here. And then they burst open to reveal these dark red, orange, or even sometimes yellow seeds. There's these trees on Molokai that have yellow seeds. They're really cool. So the name and the visual of the twisting pods are occasionally referenced in Mele and Mo'olelo as metaphors for change, beauty, or complexity. And the natural phenomenon is symbolic of that cyclical transformation, right? And rhythmic energy that is especially accentuated in nature here in the islands, right? So Wiliwili trees are also described in Mo'olelo as indicators of seasonal changes like we just saw. The bright flowers and seeds are tied to observations of reproduction and dispersal, especially where they stand out in those dry forest areas, right? So there's no like singular myth or story about the seed pods themselves. It's just the twisting pods are a narrative that's embedded in its name, which reflects a Hawaiian tradition of encoding that natural behavior into plant nomenclature. So it's one of those examples of a mo'olelo expressed through observation and naming rather than a full tale. And then, of course, the surfboards, right? The tree itself has twisted bark and twists all around the environment too. You can see in this top left picture here, or top right picture actually, we dubbed this the octawilly. So they branch out, right? They're trying to increase their surface area to funnel more rain to its main trunk. And so like throughout my study, one characteristic of a dry forest tree making the most of its habitat was to literally branch out in as many places as you could. Whereas the exact same species in an environment where there are abundant resources, like at Kalailoa, there's this underground water feature, or in Kauai's rainforests, or at Cocoa Crater. I live right next to Cocoa Crater, and there's irrigation there, and those trees grow straight up to the sky. There's no branching out. But anyway, the wood itself, it's very lightweight. So it's unlike the seeds too, right? That's how you tell a native erythrina from a non-native. Did you guys know that? The Wiliwili seeds will sink in water, and the congeners, others in the genus, they'll float. So that's how you can tell a Wiliwili seed. So the wood floats, the seeds sink. So I mean, just imagine like how many uses there would be for a lightweight buoyant material back in the day, right? I'm thinking like fishnet floats, outrigger canoe amas, anything you don't want to sink in the ocean, really. Okay, moving on. So back to the background here. So that plasticity that I showed you in the trunk, that's also seen in the corolla color as well, and in the seeds. Sometimes even on the same tree though with the flowers. So in the bottom left corner there, that's one tree at Cocoa Crater, two different flower colors on the exact same tree. That's highly unusual. And just the variety of floral colors in the species is unique to Wiliwili of all the other congeners. So the genus Erythrina is thought to be bird adapted for pollination, from hummingbirds to passerines. Other visitors have been documented as well, and those include bees, other insects, and even bats. But because of the early adaptation in its evolutionary history, the genus is largely thought to be self-incompatible. And a lot was known about the genus when I was getting into this. Nothing was known, however, about the reproductive ecology of Wiliwili specifically. There were possible contemporary pollinators that were reported anecdotally, but nothing was published for certain. Okay, so we know the threats that exist in these ecosystems, right? Invasive grasses, fires, insects, oh my. But on top of these, there may be these underlying factors directly limiting recruitment that we don't know about, right? And across Oahu, I saw very little regeneration occurring. So we needed a basic understanding of these fundamental ecological aspects that hadn't been identified for Wiliwili to be able to understand what we want to see in response to threat control, right? If we want to control for grass, we can't look for seedlings if there's no seeds being produced, and we can't look for germination if there's an inbreeding depression occurring. So I had a lot of questions, but I limited to 4 that focused on the life history biology that wasn't quite understood yet for Wiliwili. Here they are. I asked, what are the current visitors? Who's visiting the flowers? And then what is the effect of pollination treatments on the seeds and seedlings? And then do they also vary across sites? What about recruitment? Where is it happening? And limited to Oahu and Big Island. And then the genetics, but I won't bore you with that part. Please reach out to me if you're interested. It is a really cool study, but might be really boring for this. All right, so first was the pollination biology study. And these are the methods. Really, the thing to pull out here is I focused on 3 locations, 1 garden site, 1 wild site, and 1 preserve. That's Waikoloa. So here are the methods. It's just know that 2 were on Oahu, 1 was on Big Island, and what I mentioned before, garden, wild, and preserve. So here is what we found. These are the avian visitors, and they included the house finch, bulbuls, Japanese white-eyes. There were myna birds, red-headed cardinals, and more white-eyes. So notice in this picture here, the white-eyes are small enough that they can approach from behind, and they can even clutch that banner petal and really do a lot of damage in the process. And then they rob nectar while they're doing that. But the larger birds, they're not agile enough to do that. So they kind of clumsily hop around on the tops of the inflorescences, brushing both the male and female reproductive parts as they kind of hop along. But I didn't observe any preference of corolla color. At first, I was thinking maybe one of the flowers would be more attractive to some avian visitors, but I didn't notice any difference. They all go for all of the flowers, and if they can see that are open. All right. And then we have the insect visitors. So these included paper wasps, honeybees, the small emerald carpenter bee there in the bottom corner. And that's a non-native Hylaeus, the yellow-faced bee, furiously collecting pollen. They are very good at what they do. And then the honeybee there, not pictured in any of these were the carpenter bee, the saffron finch, and then these yellow crazy ants that were insane nectar robbers. And they also farm a number of sucking insects on the plant. So they're bad news. And then John Price and a student of his on the Big Island looked at bill and floral morphology to determine if there was a past pollinator of Wiliwili. And they found these three native birds kind of fit the bill. So EEV, you know, the range doesn't really overlap any longer, and the Oahu o'o is extinct now. But Amakihi maybe potentially could still pollinate at lower elevations. I didn't witness that, but it's possible. What I did witness currently is that the myna, the carpenter bee, and other large birds could be pollinating. Definitely the white eyes, honeybees, and wasps on nectar robbing, and then the pollen thieves were the hylaeus and the honeybee. So also, I wanted to see if there were any pollination syndrome evident in Wiliwili. So I looked at nectar sucrose concentration. And for the nectar study, I focused my research on one site only, and that was the Cocoa Crater Botanical Garden. Main thing to note here is that I only conducted this part of the study at Cocoa Crater. And we found a mean average, that is 14%. So it's dilute. It's a large amount of dilute nectar, and that's very indicative of an Ornithophilus pollination syndrome, which is consistent with what we just saw, right? Birds, large passerine birds. So the Hylaeus too will collect nectar, but it's really too dilute for the bee since it's not co-evolved for that mutualism. But we did find a Hylaeus trying to ingest nectar. That was pretty cute. Okay, so why does it matter who's pollinating the trees? Well, as we just saw, some are able to rob nectar. So are these possible pollinators really doing service to the trees? That's why I went to this next part of the study. This was the pollination treatment study. I focused on two locations, one garden site, one wild site. And key to understanding some of this is knowing that the garden site is irrigated and managed for grass. Mokuakea'au was not. Okay, so I had two controls. The first control was flowers just left open to natural pollination, no manipulation by me at all. But the second control was a cut inflorescence, and that was to account for resource allocation issues. And I'll go into that more in the next slide. But then we had an autogamous treatment where the flowers were bagged, reproductive parts were not manipulated at all. And then we had a getonogamous treatment that was where flowers were treated with pollen from a different flower on the same tree. And then the xenogamous or cross is what we call it. Flowers were crossed with pollen from a flower on a separate tree more than 20 meters away. And then this explains why we had the two controls. So it was research done by Medrano et al. In 2000 that showed acropetalous inflorescences, like Wiliwili, usually allocate resources to early proximal flowers. So to account for those resource issues and the potential for cross-contamination of pollen from the remaining opening flowers, it was determined that we should trim the remaining inflorescence. All right, so we bagged them in bud form. The getonogamous and cross treatments were emasculated and then pollinated following literature protocols for each treatment. The inflorescence was clipped, treatment done, hanger on, and then we bagged them. So those contraptions there are wire hangers that we had to create to kind of protect the flower and our experiment. We even tied the hangers and bag to adjacent branches to avoid that same damage and also eliminate any chance of pollen being rubbed off from the bags, kind of floating around in the breeze. And then seeds were harvested about 1 month after treatment. And the results were, oh, that's me pollinating a stigma there. Okay, so these were the results. All pollinations here, treatments here were compared to the second control, so that's important. That's the one with the cut inflorescence. So there were significantly more fruit produced per flower in the cross treatment compared to the control and other treatments, but it did not vary across sites. For seed set, there were more seeds produced per flower in the cross treatment too. So you can see from this graph also that Wiliwili flowers are pollen limited, with the control setting far less seed than the cross treatment. So potential pollinators may not be so effective. All right, on to mass. There was no significant difference across treatments, but seeds from Makuakea'au were significantly lighter. Germination, so this is the probability of germination from treatment to treatment. The y-axis is the percent of total seeds that germinated, and there's the sample size on the top of the bar. So I was really limited by sample size here. With a higher sample, we may have seen a statistical difference, but no significant difference noticed across treatments. However, slightly less germinated from Makuakea'au. So then, when no significance was found, I was curious what other factor could be contributing to seedling vigor. And further analysis revealed that mass was lighter at Makua and germinated even less, even when mass was held constant. So something unknown was occurring there. We still don't know what that was, but future research could dive into that. Okay, there was no significant difference across treatments or sites for the time that it took to germinate, but the average time was about 6.5 days. And then the cross treatment was a significant predictor of final seedling height compared to the control and other treatments, but site was not significant in this particular analysis. And then I also got curious if the seed mass was a predictor of final seedling height, and yes, it was. As mass increased, so did the height of the seedling. So neither site nor treatment was significant in predicting the final total leaf area, but the cross treatment was a significant predictor in the final basal diameter of the seedlings compared to the control and the other treatments. Okay, so here is the summary there of significant findings. And if you want all the details, this chapter was published in Pacific Science. It is behind a paywall, but if you want to email me, I will send it to you happily. Okay, so on to the demographics, because in addition to the findings that I just showed you, I wanted to see if Wiliwili was naturally regenerating in the field. I just showed that seeds are being set, so are they establishing? Okay, so the demographic study took place on 2 islands and 8 populations, all listed here. It occurred at every field site in the last slide. And I recorded these biotic and abiotic factors listed that influence regeneration. So that included the bruchid beetle, gall wasp infestation, canopy cover, grass, all that. And these are the results. So the number of individuals is on the y-axis and the life cycle stage is on the x. What do you see? There's nothing regenerating. The only place where we saw a fair amount of recruitment was in the garden and a few in the Waikoloa Preserve too. But the wild sites are just not regenerating at all. There's some obvious bottlenecks in recruitment here. The first in the seed to seedling transition, but then also in the seedling to sapling transition. And here, it's really small, sorry, but you can kind of see some of the factors that are contributing to the lack of recruitment. I didn't have enough populations to do statistical analyses, but if you look at these factors in the populations that have some regeneration occurring, it looks like fencing and grass control allow for some amount of recruitment. So you can see here how bad the galling was at Waikoloa. This is all at Waikoloa Dry Forest. And the probability of seeds germinating decreases as the galling increases. The bruchid beetle too, so Kaufman in 2014 found that seeds that had 1 bruchid beetle hole was reduced to less than 10% germination, and seeds that had more than 2 bruchid beetle exit holes, they didn't germinate at all. Excuse me. And the longer that an open seed pod stays on the tree, it's the greater probability that the seed will be infested with the beetle larva. I found you have about 2 weeks. They say to harvest the seeds within a week. I think after 2 weeks is when yeah, the bruchid infestation is so bad that the harvest would take a long time to pull those out. So anyway, yeah, and we have a biocontrol for the bruchid beetle. It was released over 10 years ago now, but it's still really bad. All right, in summary, non-native species are potentially acting as pollinators of Wiliwili flowers. So they're clearly setting seed at the top of the trees, but it's pollen limited, right? So that's common in self-incompatible species, but contemporary visitors are probably not so effective as pollinators. The species is an outcrosser, but not obligately so, since seeds were set under selfing conditions, and so that indicates a mixed mating system. The heavier seeds we observed at Cocoa Crater, that may be tied to the irrigation, but in the context of climate change, lighter seeds could be real problematic for natural regeneration. And then the bottleneck. So it could be the bottleneck and the lower seed output, potentially effects from the gall wasp and the bruchid beetle, but also because of the lack of cross-pollinators, right, that allow for cross-pollination. Even more concerning, though, I think, is the seedling to sapling transition, which that may be due to grass cover and ungulates, but it may also be related to drought. So I show that seed mass is a driver of germination probability and seedling height, and larger mass, that may be correlated with environmental variability in water. So with drier years ahead, we may even see less recruitment. Based on my population structure and genetic research that I did not present here today, populations are isolated and fragmented. If there are more individuals in a population, there's a greater chance of cross-pollination. And we saw that that increases fruit and seed set and correlates with more vigorous seedlings. Recruitment was only observed in areas that were fenced and or managed for invasive grasses. And again, with the drying climate and bottlenecks in recruitment, it may be beneficial to start planting Wiliwili at wetter climates. I suggest that future research should probably look at seed germination and survival in wetter climatic conditions, possibly at higher elevations. And then the IUCN publication, that status was vulnerable in 2018, but that's going to need to be updated in 10 years. So 2028, if we have any IUCN assessors in the audience. All right, beyond its historical uses and significance in the Hawaiian culture, excuse me, Wiliwili is now a focal point in modern ecological restoration. So there are tons of organizations and conservation groups like Waikoloa Dry Forest Initiative and Protect and Preserve Hawaii that are actively replanting Wiliwili and degraded dry forests. So those efforts go hand in hand with cultural education programs that reconnect communities, especially the youth. With knowledge of how Native Hawaiians revered and utilized the tree, it's schools, it's local nonprofits, community events. They're all integrating traditional ecological knowledge like lei making and mo'olelo storytelling, and mixing that in with hands-on restoration, reinforcing that connection between people and place. In fact, the Waikoloa Dry Forest Initiative holds a community work day. They're Hui work days, I think they're called, and they have a Wiliwili festival every year. That's that Wiliwili fun picture down there. So there's this revival of traditional knowledge and stewardship occurring all across the island. It's really, really exciting. So, it's that deep connection, right? It's between Hawaiian culture and the Aina. The ethnobotany of Wiliwili is really key to biocultural restoration because it has never solely been about restoring the natural environment, right? It's also about strengthening the cultural relationships that people have with the Aina and revitalizing the culture right alongside the land. I think to truly conserve Wiliwili, we must aim to restore both the biophysical elements of an ecosystem and the cultural knowledge, traditions, and relationships that sustain it. All right, I'd be remiss if I didn't mention my company. This is Terraformation. They've been doing great work on the Big Island of Hawaii. This site is Kaupaloa, and it's a highly degraded former Iliahi forest, actually. We've planted over 35 acres there with 22 native species. We're also doing a lot of seed banking with Wiliwili. They are candidates for long-term storage. We have TerraWare. It's our own plant tracking software, tracks seeds from the seed bank to the field. Outreach, always important. And alongside this, capacity building is even more important, especially with the next generation. Passing down that cultural knowledge is probably one of the most significant ways you can honor the Aina and all who came before us. That's all, folks. So these are the amazing people who facilitated the project. Special shout out to my former lab mates who followed me up into the dry jungle with wire hangers, mesh bags, and a great attitude in 100-degree heat. And my committee, especially Tamara Tickton, she was a chair, fearless lab leader. And then a special thanks to Christine and Wes, who's behind the scenes, and Gian from Honouliuli, who recruited me and have been awesome to work with. Also, thanks to anyone here who's working with conserving and or preserving Wiliwili today. I know it's the labor of love. And with that, I'll take any questions. Thank you very much. Oh, and that's my email down there if you'd like to ask me any questions over email. And I'll stop sharing now too.
So that was great, Emily.
Oh, thanks.
Maybe as we're letting folks maybe think about questions, I really was a part in syncing. That's a neat factoid. And yes, I would like to request a copy of your publication as well.
Sure.
So I have two questions maybe to start us off with, as maybe people are thinking through what you shared and developing their own questions for you. But I guess the first question is just, what for you was surprising as you started your research and your findings, and kind of what were things for you that stood out that was just kind of a surprising conclusion, or just something neat that you didn't think you'd learn about?
I mean, the whole thing, right? Like, I think I wanted to know what was directly affecting this tree that I loved, but I found out so many cool things along the way. I didn't like at the time that I started the study, I had noticed, the galling effect and things like this, but I didn't know that we had a biocontrol, and I didn't know that it had been released just a few years before I started my research. And so it was really, actually a great time to get out in the field and do these kinds of studies that measure infestation. I wish I could have done more populations. And that's, I think that's probably my biggest regret of the study is but you only have 2 years. I wish I could have had, 60 populations, but yeah, that costs a lot too for a grad student traveling. There was also a part in the genetic study when there were kind of anecdotal reports of seeds that were kind of transferred from the Kohala area of the big coast of the Big Island on the northern Kohala coast there. That seeds had been given to Kaho'olawe. And then when I did the genetic research, the Kaho'olawe and that particular population, they were a genetic match. So that was really cool to kind of prove anecdotal evidence. Yeah. And then I didn't know that Wiliwili was used medicinally either when I first started. So and a lot of the ethnobotany. Yeah.
Hey, Emily, Christine, we actually have a question from Christy Mello. I'm gonna unmute her right now so she can actually ask it directly. Christy?
Oh yeah, thank you, Emily, for your presentation. I enjoyed your presentation today. I had
thanks.
Question. So for repopulating the Wiliwili, one of your conclusions is that there's a need for wetter climates and perhaps higher elevation. I know with a lot of native bird species in general are greater losses happening at the lower elevations. Is there a relationship between them as populators and needing to go higher up?
Yep. Yeah, exactly. Yeah. Right. That I think that slide, it shows the EEV. I don't know what EEV's former range was. So we don't know if it was a pollinator of Wiliwili or not for sure, but now the ranges don't overlap at all, right? It's been pushed up into higher elevations and Wiliwili is still at lower elevations. Yeah.
Thank you.
Oh, thank you. Good question.
Thank you, Christy. And then I have David. See if I can unmute you right here. Mr. David Johnson, go right ahead. If you'd like to ask it directly.
Oh, okay. This is Beth Flynn. I'm watching with David. Thank you for the wealth of information, Emily. That was really interesting. Is there any role that citizens could take in using Wiliwili in landscaping their own yards that might help in the species conservation?
I definitely am constantly propagating Wiliwili from the seeds that I find. There's a just down the road from us, a guy planted his whole, the median in between the sidewalk and the street. He planted Wiliwili all in there. I have one, I wish we could plant more in our yard, but I don't think our yard could handle it. But I did plant one in our yard. Yeah, I think that's a great way to connect with the tree. And it's a good way to learn how to propagate it too. I was actually going to show you guys how to propagate Wiliwili on this, but I couldn't find my trimmers in time. But if you want to know, you just nick the seed coat. So there's a top and a bottom to the Wiliwili seed. The bottom has like a circle on the bottom. You can tell it's kind of like a popcorn seed. You've seen the popcorn seed? You can tell there's a top and a bottom. So the top of it, there's a seed coat that kind of extends beyond the embryo and the seed, and you just nick the top of it and then soak the seed in water overnight, and then you plant it in the morning, and not very far in soil, just tiny bit in the soil, and they pop up. Super easy to germinate too. It's like 86% germination rate. They're really hardy.
Wow. Nice.
Yeah. And of course, email me if you have any questions. But that Native Plants Hawaii book has all the propagation techniques in there.
Outstanding.
Yeah, I love that book. Good question. Thank you.
Yeah, thank you. I mean, I'm waiting to see if anyone else has any other questions. They may have directed them directly to you during your chat. Or if Christina's received any as well too, because I said it so it's direct message to us. So
yes, good call. Nick, hi. Quickly summarize the genetic investigations. Geez, and Mitsuko's on this call. It's like pressure is on. Quickly summarize. Yeah, the populations are all very isolated and fragment. There was literally almost zero admixture in between the populations, distinct on islands. So you could tell the Big Island populations were very distinct from the Maui Nui. So that's a good point. The islands of Maui, Lanai, Molokai, and Kaho'olawe, they were all, or not Kaho'olawe, no, but the Maui Nui islands, they were all the same genetic structure. And then separate for Big Island and Kauai. I hope that makes sense. But email me if you want to know more, and I will send you that chapter too if you want. And then there's another question from Antonia. Did you say Japanese white eyes are not good cross-pollinators? In our observations, that's what we noticed. They barely ever contact both reproductive parts because they can sneak in from behind. They're so small that they can either avoid the stamen and stigma by reaching around it, or they can clutch the banner petal and just dive right in the banner and heel petals and grab the nectar without touching any of the reproductive parts. So they're pretty conniving. But I know that they are pollinators of other native plants, and actually effective pollinators of other native plants. I cannot remember what those are right now. Sorry about that. I hope that answered your question, but yeah, let me know and email me if that wasn't clear.
yeah, thank you, Emily. And then I don't know if you put your email in the chat. Did I?
I will do that.
Yeah, just for everyone in case they want. And then I think that's everything that I was able to see. I don't know if Christine's seen anything else, but we also don't want to take up any more of your valuable time. We appreciate you presenting.
Oh no, this is great.
No, thank you, Emily. We really appreciate it.
Thank you.
Yeah, likewise, my thanks as well. And I'll just kind of wrap up with a couple of notes for folks. If you do want to learn more about the actual park itself and updates and some of the history, again, we do have a couple of the series or speaker series that we have enjoyed with some of their other speakers, and I'll put that in the chat as well. Let me just change my settings to everyone in meeting. We have our premier archaeologists who did a lot of seminal work, at the park, and I also gave a talk to give the latest update on what's happening at the park. So feel free to check those other speakers out as well. And just wanted to give a quick preview of things that you can look forward to in June as well. So we are dedicating, actually, Emily, it's great you mentioned Kalailoa Heritage Park, because they're actually going to be one of our speakers. So the month of June, we're dedicating to the Honouliuli Ahupua'a and all the wonderful work that our partners are doing in the Ahupua'a. And kind of continuing the theme of cultural and natural resources, we're going to have Pauline Sato with Malama Learning Center, as well as Kavika, who is the park manager at Kalailoa Heritage Park. So they're going to be speaking, our virtual speakers for June, June 3rd at 5 p.m. And you can register for their talk on the link that I just put in the chat for all our June events. In addition, Malama Learning Center is going to host us on one of their work days. So you'll find registration for that as well for June 14th. And finally, in June, we're going to have a free screening of a film, Untold Stories, that features Honouliuli. It's a local production company that did a documentary. And it's going to be thanks to Pacific Historic Parks, who's sponsoring the film series at Doris Duke Theater at the Homa. It's going to be free, but you do have to register for the tickets. And at the end of that our documentary, we are going to have panelists with the producer, one of the descendants whose grandfather was held at Honouliuli, and as well as Carol Hayashino, who was the former Japanese Cultural Center Hawaii president and CEO, who really led the charge to help establish Honouliuli as a national park. So they will be there as part of a panelist at the end of the documentary to help answer any questions and to learn more from them. And finally, in June, we have a great pop-up exhibit for Honouliuli, going to be featured downtown at the American Institute of Architects Honolulu chapter office. And all that is slated for June, and you can find all that information in the link that I just provided in the chat. So all great things to look forward to in June, and you can see the full schedule of the rest of our commemoration year on our website as well. It's broken out by months, and so each month has a theme and a lot of great virtual and in-person opportunities to continue to learn more about the park. So with that, yeah, I definitely want to thank Emily. We learned a lot about Wiliwili and really looking forward to maybe partnering with you of the work that we could do at the park. Beth asked about how people can volunteer, and we are hoping to be able to have work groups and volunteer groups up to the park maybe next year, and maybe we can make Wiliwili restoration a focus of some of that work. So we look forward to continuing the conversation and building on what you just shared with us, Emily, for the park and our Wiliwili there. So thanks everybody again for joining on this Wednesday evening, and I hope you have a lovely rest of your evening, and we look forward to seeing you again in some future event. Mahalo, everyone. Bye.
Mahalo. Aloha.